Good morning, everybody. This is the 37th Global Online Seminar in Biodiversity Informatics. I'm Tom Peterson, your host for this series. I'm not much on introductions, as you all know, if you've seen any of these seminars before, but I'm very, very pleased to welcome our speaker today. I'll presume to call him an, let's say, informal academic grandson, because I was an informal advisor to Rob Anderson, who's gone on to be quite a leader in this field. And Jamie Kass is an advanced PhD student with Rob at the City College of New York. Jamie and Rob and their broader group have put quite a lot of development work into a package called Wallace. And I was very pleased when they contacted me to see if we could dedicate one seminar to this package. So I'm just going to leave it at that and welcome Jamie and say thank you very much, Jamie, for doing this seminar.
Thank you very much for having me. This is exciting. I'm going to try to look into the camera, but I have this enormous monitor in front of me, so you have to pardon me if I'm not looking directly into it at all times. So let me start the presentation. Okay, so please send all questions to this email address, and I'll try to take them at the end of the presentation. So the title of the talk is Wallace, a flexible platform for reproducible modeling of species niches and distributions built for community expansion. This is our current development team, and these are all of our numerous affiliations. So a bit about me, very briefly. So I am a PhD candidate in Rob Anderson's lab at CCNY. We do biogeography, a lot of spatial modeling. My dissertation is on integrating biotic interactions into distribution/niche models. And I'll be beginning a postdoc next year at the Okinawa Institute of Science and Technology in Okinawa, Japan, in the lab of Evan Economo, who's also a biogeographer and who works with ants. So the ubiquitous slide about species distribution/niche models. I'll go over this quickly because this might be a review for a lot of you, but might not for some others. So what we'll call distribution models are models that use occurrence records of species and environmental data. We take the values of the environmental data at the coordinates of the occurrence records. We use an algorithm to build a relationship between those two things. And we have a model of what we're going to call a niche, which is more or less usually an abiotic representation of the niche, because we're mostly using climatic variables. This is not the case at all times. And then we use that model to project suitability onto a map, because if we have environmental gridded data for an area of interest, we can use that mathematical model to predict what the suitability will be. And so you'll see that in the map over there. It's going to be some kind of continuous gradient from non-suitable to suitable. And the interpretation of suitable can get a little bit hairy, but we're just going to call them suitable areas for now. So traditionally, these kinds of models have been built using some kind of GUI. A lot of researchers were using code to build models, but because a lot of easy-to-use GUIs came out, most researchers ended up shifting to GUIs. The example here is a very poor resolution image of the Maxent GUI. It's just one example of a number of GUIs used to make these models. These GUIs often have default settings, which are supposed to be generally useful in a general case, but might not be appropriate for your particular case. They're relatively inflexible. They usually have settings, some of which are a bit hidden. And if you're not good at exploring the GUI, you might not find a lot of the settings or know that they exist. They're also infrequently updated. So these GUIs are the product of a single individual or a research team. If that individual or team does not have the time to update frequently, it doesn't get updated. And then they usually require numerous other applications in order to complete a full modeling analysis, because you need to format your occurrence data using some kind of spreadsheet software like Microsoft Excel. You're going to need some kind of mapping software like a GIS in order to view the maps and make maps after you build the model. And so a lot of different software packages are needed in order to complete this. On the other hand, we have code. Code is written by an individual, so it is inherently error prone. If you make a mistake in your code and it doesn't make an error, that little mistake can get perpetuated through the entire analysis, and you wouldn't even really know unless you viewed it more carefully the next time. This is more hard to do with the GUI because of its inflexibility. It's kind of built so that you won't make so many huge mistakes. And the code also is not usually easily generalizable. So usually you're writing it for yourself or for your particular project, and you don't usually comment as well as you should. And if you were to hand that code off to somebody else, they might not know how to use it or how to commandeer it for their particular purposes. So each of these things has positives and negatives. So there is a need in biodiversity informatics for software that achieves this balance between automation and supervision, particularly for some kind of software that would automate the repetitive aspects of coding. So unless you're a whiz at for loops, a lot of coding is pretty repetitive. And even if you are a whiz at for loops and you know what for loops are, there's a lot of repetitive aspects to this too. So GUIs can take care of that for you. It also, we're interested in developing software that forces users to make decisions. So if you're an ecologist, it would be best if you wear your ecologist hat while you're doing your analysis instead of just pushing buttons. And also, it would be good to have some kind of software that is general with respect to all the different algorithms that are used to make these models. There's a wide diversity of algorithms available. Some GUIs exist that offer all the algorithms you could ever imagine in the software. But again, all of them usually have some kind of default settings and are hard to change. So our team had an idea to combine code-based methods with a GUI. So a lot of, or most of, the new methods in ecology these days are released as code in some form, usually as R packages in ecology. If you pick up any issue of methods in ecology and evolution, you'll see every month there are 3 or 4 new packages that come out that are very useful for a particular kind of analysis. However, if you don't really know how to use code or are not comfortable using code for your analysis, they're not really accessible to you. So our idea was to have a GUI where the foreground is this interface, kind of like what I just showed you, but that the interface, our idea was that the interface should be flexible, it should be extensible, so when you want to build new parts of it, it should be easy to insert those new parts in for when new methods come out. And it should also be informative. It should have information about the methodology inside the GUI to teach you about the methods as you're using the GUI. In the background would be R code. Why R? Because R is the lingua franca of ecology right now. It is not necessarily better than any other language. It just happens to be what everyone uses, and I find it quite useful. So R would be running because there are so many existing and new packages in R that allow people to do great analysis in ecology. Our team came out with some recent-ish R packages, namely SP-Thin, which does spatial thinning of occurrence points to reduce spatial bias and spatial autocorrelation, and also ENM-Eval, which iterates model building in Maxent and allows you to do model selection. And so we wanted to disseminate the methods in these packages to a broader audience than just the ecologists who code all the time. So we submitted our idea, we called it Wallace, to the Ebbe Nielsen Challenge, which was put on by GBIF, and the call was to develop a project that makes innovative use of GBIF data. And we were lucky enough to get to the finalist round. What we did was we combined these methods that I just discussed with an R package called Shiny, which allows you to make a kind of like a web app style GUI that has R running in the background, which is exactly what we wanted to do. So we're thankful that Shiny existed when we were thinking about this. And what we came up with was this, Wallace. It's the software that I'm talking about today. I should mention that it is named after Alfred Russel Wallace, the founder of biogeography and co-discoverer of evolution, who is often overlooked, but not anymore. So I'll talk a bit about what is Wallace and why do we think Wallace is important. There are some qualities of the software Wallace that we think exemplify what modern scientific software should represent. So in a sentence, it's a point-and-click application, which is a GUI, a graphic user interface, that features a niche/distribution modeling workflow. But we envision that in the future, it will include many more ecological modeling techniques. So as I mentioned, Wallace has certain qualities that make it great. They are listed below. I'll be going over each of them briefly. I just wanted to mention there's a link underneath there for the Wallace webpage, which will be at the end of the demonstration as well. And this is the original development team that came up with the software, and we published a software note recently in Methods in Ecology and Evolution. So please give it a look if you're interested. So first of all, Wallace's code is open and it is free. So that means that the code is up on GitHub, which is kind of like a Facebook for nerdy coders. And you can go there and view, download the code, edit it, do whatever you want with it anytime you want. We think that this is very important going forward because this epitomizes flexibility, and it also allows people to play around with what exists and alter things for their own uses. I should also mention that Wallace makes use of open databases like GBIF and VertNet for bioinformatics. And so we allow access to these open databases as well. Wallace also provides guidance, so it's instructive. There are 2 levels of guidance, and I'll get to that in a bit. And this guidance addresses conceptual and methodological issues, and also includes links to the literature, which we thought was important. Wallace is flexible, so it provides the user with multiple options, and also allows different kinds of interactions with the data that you're analyzing. So you can input data to Wallace, and you can export data from Wallace in multiple forms. And Wallace is interactive. So the focus is a huge interactive map that kind of feels like Google Maps that you can use to explore your points and take a look at the area of interest on a dynamic map. But it also has dynamic tables that you can sort And it also has dynamic graphs that update when the user changes various controls. And Wallace is reproducible. So this was especially exciting. So we were able to program Wallace so that at any point during the analysis, you can download a script that is annotated that you can run in R to reproduce all of the results in your session. And I'll talk more about that soon. And finally, Wallace is built to be expandable. So we know that for each of these steps, which I'll outline in a bit, of the analysis, there are many different options that different people have advocated and different labs talk about and want people to use. And we want to make sure that these people can add their methods into Wallace. We're interested in plurality of science and not advocating for a particular way of doing things. And so we made Wallace modular, which means that the analysis steps consist of different modules that the user can choose from. And I'll go over those too. So I'm going to begin a walkthrough of Wallace. And let me now, before I do that, I have a few things I want to say. So first of all, how do you install Wallace? For those of you who are not R users, it might be a little mysterious, so I'll go over this very briefly. First, we need to install R, which is free, so don't worry about that. And optionally, RStudio, which is a big wrapper for R that I find very, very useful for programming and has a lot of neat features that make R a bit easier to use, but it's not necessary. You would, if you want to use Maxent in Wallace, you would need to download it. I have a link right there to the American Museum of Natural History, which is currently housing Maxent. Maxent is now open if you did not know. And so it's also easier to get. You also need to make sure you have the appropriate version of Java for your system. Maxent runs on Java. There are sometimes problems with installing Java, and I have troubleshooting on the GitHub page on the link below. So if you're having any problems with that, I've encountered almost all of them and written how to fix them down on the GitHub page. So please see that. And then you'd also need to place the maxent.jar, which is the software that runs Maxent, into the folder for the Dismo package. If this all sounds like mumbo jumbo to you, then as you're using Wallace, it will inform you exactly where to put it. And it's really not a big deal. You just kind of move a file from one place to another. Unfortunately, Dismo works this way. I'm not very happy about it, but there's not much we can do. And we're thinking about ways to make it a bit easier in the future. However, if you don't want to use Maxent, you don't have to bother with this at all. You install the Wallace package from CRAN, which is a R package repository with these 3 lines of code. Literally, these 3 lines of code, once you run them, Wallace will be running unless you encounter enormous problems. And then you should contact me. So before I jump to the live demonstration, Wallace is composed of components, which are analysis steps. We've broken down the analysis into 8 steps. There are certainly, people could argue that there are more than these. We tried to simplify. We might include others in the future, but for now, there are these simple 8. Within each of these components, there are choices, and those choices are called modules. So in the A row in the diagram, you'll see the components. It starts with obtaining occurrence data, obtaining environmental data, processing these, and it ends with modeling and then visualizing the results of the model, and then also projecting it to other areas and times. The last column is the session code column. So this is where you can download your code. And you can see there are a couple of options for file formats for downloading the code. The B row are the upload and download options that currently exist for each component. The C row are the major R packages that are used. So we made this big effort to try to highlight the R packages that are used in each of the components. Wallace is kind of this big application that uses a lot of packages, and we wanted to make sure that we acknowledge all the working parts. And then D is the modules, and I'll go over a lot of them during the live demonstration. And now we begin the live demonstration. So I'm going to open up our studio, and I will type, it's probably hard to see this. I will type library Wallace, which loads the Wallace package. I've already installed it. R will load up these essential packages, Shiny, which is what I just discussed for the web app interface, and Leaflet, which is the map software that Wallace uses to make the map. And then I will use the only function in Wallace that is accessible to you, and that is run Wallace. And there are no parameters, and I press enter, and then I get Wallace. It's literally that easy. So even if you're not an R user, it's quite easy to do this if you have R. So this is the landing page. It explains what Wallace is, who we think it's for, attributes, which I just discussed, a link to the email, some references. This is just the workflow. So we start with occurrence data, and you'll notice that there are two main parts to the screen. There's this left side and there's this right side. So the left side are the controls, and it's also where you choose which module you're on. Up here are all the components. So we're on the first component, which is obtain occurrence data. And we're currently on the module called query database, which allows you to download occurrence records from different databases. You'll see the map is over here. We can zoom around on it like a Google map. You have little zoom controls. And then these other tabs are not relevant now because nothing's going on, but besides components, guidance, and module guidance. But the table and results are just not there yet. Before we use any of these controls, it's a good idea to kind of read the guidance. We tried to keep it as brief as possible while also being informative as possible. And again, we included references in case you wanted to know more details. So the component level guidance is about obtaining the occurrence records themselves. And the module guidance is specific to each module. So when we are on the query database module, we see information about these databases, but when we're on user specified, we would see a different guidance for this module. So you can enter your own data as a CSV in addition to getting data from the databases. So what we'll do is download some data from GBIF. I will download data for Tremarctos or Natus, which is the spectacled bear. And I will leave it at the default of 100 occurrences. So I'll press query database. You'll notice that in the corner it has a little progress bar, and we get our data. You'll notice that some of these points might be a bit dubious, but we'll address that soon. And we can zoom in and take a look at where a bulk of these points are. They're in and around the Andes Mountains, which is a good sign. Thank you, GBIF. And you can also download these data using this button. So we can press this, we can open that in Excel, and we can see that we have the entire GBIF table for these 100 records. It's quite long. Because it's so long, we decided to condense it a bit and show you this, this condensed version of the table, which includes the columns we thought are most useful to biogeographers. We might add some columns in the future, but these are the ones we thought were most important right now. We also added a unique occurrence ID because as we will be modifying this data set, you might want to know which points were removed during the cleaning. You'll notice that up here there's a log box. So this tells you after you complete any kind of operation, it will report the results to you. So it tells us that we got 100 records out of GBIF, but there is a total of 237 in the database. If we want to get all the data possible, we might want to increase this range a bit. Some records were removed, so 51 records had no coordinates. Those were taken out. And there were 3 duplicate records, which means the lat longs were exactly the same. So the remaining records are 46. We have 46 data points right now. This is a very basic cleaning step, but is very important because you don't want to include duplicate data. So we will now move on to the next step. I also want to highlight that you could change the base map up here. So if you don't like the Esri topo, you can look at the Nat Geo base map, or this one. There's a lot of choices. We're going to stick with this because it's the easiest to see. So we'll move on to the next component, which is processing your occurrence data. So as we saw, there are some points that are in areas that I didn't know spectacled bears live in, like southern Japan and LA. So those are pretty dubious. And I'm not a bear biologist, but still, I'm skeptical. So because I'm so skeptical, I might want to reduce my data set spatially to just the area that I'm sure that the bear lives in. These are probably museum records. So what I can do is draw on the map to indicate the points that I want to keep. So I click on this draw polygon button, and I can just draw a polygon around my points. I will finish my shape, and I will select these occurrences, and it zooms into them, and you notice that the other ones are now gone. So those are now out of my analysis, and I'm just concentrating on these points. I can always reset this and kind of go back to where I was if I made a mistake, but I'll keep these points for now. Okay. I might also want to remove individual points without having to draw anything. So I might zoom in and say, you know, I don't know about this point in Panama. I'm very skeptical about this. I'd click on it. It has the ID of 87. As an expert bear biologist, I could say, oh, I know that, you know, spectacled bear does not live on the Caribbean coast. I'm going to take this out. It has number 87. I enter it here, 87. I remove it, and now it's gone. And you can even see this in the occurrence table. If you were to look all the way down at 87, it's no longer there. I might also want to spatially thin. So you might have noticed that some of the points are pretty clustered in some areas. That's a problem for modeling because it induces spatial autocorrelation in the model response. And it also might be a signal of sampling bias. You might want to reduce that by thinning spatially. So we're going to use 10 kilometers as our thinning distance. So when we run this algorithm, which is SPThin, the R package I talked about that our lab helped develop, it ensures that no points are 10 kilometers, are a distance of 10 kilometers or closer to each other. So it reduces this clustering. So I'm going to run this algorithm. You'll see the progress bar. Because I'm on a fast computer, it was so quick. It's hard to see which ones were removed, so we can zoom in here. And you'll notice that the blue points were removed, the red were retained, so we can see that some of this clustering is now gone, which is a good thing for us. So now we can move on. We can also download this process data set here. It's not the same table as before, but it includes the essential information for us, which is the ID, which links us back to this table. And we notice that now we have 35 points instead of the original, I think it was 46. Yes. So now we're down to 35, but we have a much cleaner occurrence data set. So we're happy. We're going to move on now to the environmental data component. We have two options. We have getting the WorldClim, BioClim variables, which are very popular. We also have user specified, so you can enter your own rasters in, actually. What we'll do today is work with the 10 arc-minute BioClim data, which is the coarsest data, but runs the fastest. And you'll see this is just direct output from R, which is our stats on the all the rasters we just downloaded, and indicates that we now have them available for the analysis. If you were interested in selecting variables for your analysis, you could specify exactly which ones to download and use. For now, we'll just use these 19. So moving on, we are now going to process these rasters. So this is important because we need to specify the area for background sampling. So with presence background models, we're comparing the species occurrences, which are the presences, to a sample of the available area, which is known as background. And it's important to specify this correctly. You don't want to make a background that's too big or that includes areas that the species is not able to get to. This is kind of a complicated topic, and so we have some information in the guidance text, which is pretty important and you should read up on. But we have three options for defining this background, this available area for this species that we're going to compare to the presence points. We have a bounding box, which is exactly how it sounds. It's just a big box. We have a minimum convex polygon, which fits the points a bit more tightly. And we have these point buffers, which are the most conservative for the background extent. For today, we'll do a minimum convex polygon. Let's make a buffer around it of 1 degree. And we're going to sample our background points. Let's go with the default 10,000. You can increase this to however many you would like. So we're sampling right now. It's picking random cells and giving them background values, or extracting the environmental variables values for those background points. So now that we have that done, we can see that has been complete. We'll move on to the partitioning stage. So in order to evaluate our models, we use something called cross-validation. Cross-validation breaks the occurrence data into groups. It builds a model on all the groups but one, and then tests the model on that group that was left out. And it repeats that over and over until all the groups were done. This is done by assigning partition values to each point, so putting them into groups. There are two main different ways to do this. One is a spatial partition, and one is a non-spatial. So an example of the non-spatial would just be, random. Let's use 2 random groups and randomly assign a point to a group. You can change the number of folds, which is the parlance for groups. So we can use 4 groups instead of 2. We can use 10 if we wanted to, and complicate our lives. Or we can do a jackknife, which assigns each point to its own individual group. And this is good for small data sets. You can also alternatively do a spatial partition, one of which is the block. So this divides things spatially. Why might this be useful? We might be interested in building a model that can predict to areas it's unfamiliar with well. And in order to do this, a spatial partition figures this out very well, because the model, for example, will be built on these 3 groups and predicting to this last one that's left out, which is probably some kind of new environmental space. And so you have a better understanding of how well the model transfers if you were to look at the evaluation results. All of that information and more is in this guidance, so please take a look at it. For today, we're going to go with this block partition, though. We'll move on to modeling. So here's where the action happens. We can run 2 different models in Wallace currently, Bioclim and Maxent. We're going to run Bioclim first, just it's so fast. And you get your results. So we have different evaluation statistics, all of which are explained exhaustively in this quite long guidance text. So please take a look at it. And I won't go over them now, but we have different variations of AUC and emission rates, which are useful in determining how well your model did. These bins refer to the spatial partitions that we use. So we have 4 bins, and you can see how well each bin did individually, or just the average of all the bins, which are these stats. We can also run a Maxent model. So I won't go over what this means in detail. Please check the guidance again. But we can select which feature classes to use for our Maxent model. These allow differing levels of model complexity. So we're going to just look at maybe 2 of them today. And we're going to look at, so linear produces very simple models. Hinge produces quite complex models, so we'll look at both the kinds. And we're also going to look at a range of different regularization multipliers. These penalize complexity. So it's important to look at all the combinations possible of the differing levels of complexity we're interested in, build a whole bunch of models, and then look at how they all did and choose our optimal model. And so we're going to run these models. You can see that it is currently running down here. So in the background, ENM eval, that package I talked about before, is running, and it is iterating and building many models and calculating all of their evaluation statistics. So when the maxent models are done, we will see two tables, which is different from how the bioclim looks. We'll see a table that has all the averages for the partitions, and we'll also see a table that shows, like this table does, the results for each of the bins. And it's almost done here. Here we go. So this is the table that you would get in R if you ran ENM eval. You can download this table as a CSV. There it is. And you can also sort the table in Wallace. So say that we're interested in minimizing the emission rate. So we can minimize it this way. So on the top, we have the models with the lowest emission rates. You see that there are two of them. To break the tie, we might look at a different stat, like the average test AUC. We see that H2, so Hinge, with a regularization multiplier of 2, and Hinge 3 are pretty similar. I'd say probably almost the same. So let's just select H2 as our optimal model for now. What I want to emphasize is that Wallace does not choose models for you. You have to be the ecologist, you have to be the statistician, and you have to make the decisions by yourself, given with the information that's available to you, which I feel like I'm being repetitive, but it's all here. Here is the information. So we're going to go with H2 for now. So let's go to the visualize step. We can take a look at how our model did visually. So we can look at evaluation plots. They're a little bit not as interesting as they might be if you ran 1 million models, but we can see how they did on a graph. We can look at the different evaluation stats, and we see that they all did pretty similar. We have a pretty simple data set. We can take a look at the response curve. So on the X is the variable of interest, and on the Y is suitability. We can see how suitability scales with ranges of environmental variables. The variables that you're allowed to look at are the ones that have non-zero coefficients in the model. So maxent will do this thing where it tosses out variables that don't do a good job of predicting the data, and those do not enter the model. So we're only left with a subset of the original variables we chose, and so we can see how those responses were. We can also download them as PNGs. Here's a nice example. There it is. Perhaps most interestingly, we can take a look at the map prediction. Let's take a look at maxent's logistic prediction for our area of interest, our background extent. It's right here. I plotted the raster, and we can see there it is. Predicted suitability is over here on the right, and we can see that as we might expect, the high elevation areas are highly suitable. So it looks like maxent did an okay job this time. We can download the raster as any one of these things. I'm going to demonstrate GeoTIFF, which is a industry standard. I have a software called QGIS, which is a free GIS software, and I'm going to open this raster with QGIS to demonstrate how easy it is to take these predictions and move them to other software to make maps or do anything you need to do. You can even just take screenshots of this leaflet map if you want, but to make something more professional, you might want to put it in a GIS software and alter it a bit, give it a different symbology, what have you. Okay, I'm going to move on now to the project step. So we've seen how our model looks in the background extent, which is the area used for model training. We might want to project that model to other areas. So I might be interested in a hypothetical situation where spectacled bears invade southern South America. So here we can draw up a new polygon, and let's just see. Let's say spectacled bears invaded Chile. So we can draw a new polygon and project to that new area. We can see what the suitability might look like. And look, it's actually quite suitable for spectacled bears. Chileans should watch out. And we can also set thresholds. So what I didn't mention before is that we can convert this continuous raster to a binary one. So to predicted presences and absences to make some kind of predicted range map. And so here's just one of the options, but we can see if we were to make this map binary, oh my goodness, Chile will be full of spectacled bears with this threshold. But if we use a different threshold, the scenario might be a bit different. So it's a little less hazardous in this scenario. Again, we're interested in giving the user the flexibility to use lots of different thresholds. We have 2 right now. There are many more possible ones we could add, but this is mostly for demonstrative purposes. We have a very generous threshold, which I just showed you, and a more conservative one, which is right here. We could also go back to this map and look at our thresholds here. So this is what the more conservative threshold would look like if I made the map binary. You can see that blue are the predicted presences and the grays are the absences. Or we can just look at our continuous map like we did before. We could also project to new time. So for that extent, let's say I wanted to know in 2070 if the bears invaded Chile under a hellish global climate scenario. Let's just look at the non-thresholded prediction. So we can project and see what it would look like in the future. And actually, it seems to be quite good for the future. But this is an analysis that many people are interested in. Under different climate scenarios, what would the suitability look like in the future? We also might be very interested in uncertainty with this projection. So the climate in the future probably is very different from the climate today, given the scenario we did. We can calculate the similarity of the environment in the future compared to the one that we used to build the model using a MES analysis, which I just did. So the whiter colors are areas where the environment is more different in the future than it is in the present. And the darker red colors are areas that are more similar. So these areas right here that are very white probably have higher uncertainty because the environmental variables are very different in the future. So we might want to treat those with more uncertainty. Finally, we can go to our session code step and download all of our code. I'm going to do that right now. And that comes in our markdown file, which I'll show you. So we're opening that in RStudio. And here it is. It's kind of hard to see. So here is our code. It is annotated. All this is just regular text. There are code blocks in gray. That is the actual R code. You can run this in R just by pressing this button, run all. It will run this entire thing and reproduce all the data from your analysis. And you can then take it from there in R. If you wanted to make any modifications to any of these steps, say, oh, I didn't actually want to remove point number 87. I wanted to remove 88. It's as easy as doing this. Or just remove the entire line or comment it out. Do whatever you want if you are familiar with the code. The idea is that you can use this code as supplemental information for a journal article if you were to make a model in Wallace, or you could send your entire analysis to collaborators in case they wanted to run the model on their machine and see what you did. And thus ends our live demonstration. I also wanted to point out one last thing. For each of these components, we have the references for the packages that were used. So in this case, we're using the R Markdown package. We have all the authors of the package. We have a link to the web pages for the packages. And this is the same for each of the components and modules. So as I said before, we're trying to highlight the R packages and the people who made them. Okay, that ends the live demonstration. I will now move back to the presentation. Okay, so where is Wallace today? The full release, which is we were calling 1.0, came out in November of last year. The current version is 1.0.4, which really is just a couple of bug fixes, but nothing too major. The software note came out in April, or was published in April, but came out last year, early view. So please give it a read if you're interested in more detail about the Wallace project and the software. And we do have NSF funding now to respond to immediate user needs. So if users have questions, NSF can pay me to answer them via the Google group or the Wallace email, both of which are linked in the presentation. We have new exciting NSF funding to work with external partners to add new modules. So the idea is other labs that have methods that are not in here might want to add them to Wallace so that other people can use them to disseminate their methods better to a broader audience. We've already worked with a couple of partners and it's been very successful. The new 1.0.5 version with a vignette is coming soon, thanks to this grant. And we're also working on a multi-species version of Wallace where you can make multiple single species models and compare them and do things like niche overlap. And that's also thanks to our partners. And very importantly, we're also making a very big effort to broaden human diversity in our project. And we've had the great opportunity to work with a number of very smart women and minority biogeographers and coders. And we hope to work with many more. Third, we also have new NASA funding led by Mary Blair at the American Museum of Natural History. This funding is to develop new R packages that use remote sensing data and develop calculate biodiversity indicators. And the idea is to add these to Wallace and also to interface with a Colombian conservation entity, the Humboldt Institute. This is the country of Colombia, mind you, that runs a very good web app called Biomodelos. And we're working closely with the Colombian scientists to develop these biodiversity indicators and work on these packages. And that's been a great experience too. And we have the grant information and the affiliations on the right. So I'd like to thank you for allowing me to present this today. It's been a lot of fun. I hope to get lots of feedback. Here's a link to the official Wallace email. There's also a link to the Google group where if you have questions about Wallace, I'd really prefer you to post there instead of emailing me personally. There's the GitHub page, which has all the code for Wallace and all of our development stuff and everything going on. None of this is secret. It's all up on GitHub. If you have bugs and you're a GitHub user, please log these as GitHub issues. That's the most useful way for me to log the issues and record that I've taken care of them. We also have a website for Wallace, which is up there. There's a link for the Anderson Lab. I also want to mention that two other lab mates, Sarah Meenan and Gonzalo Pinillo, which are up on the top right of the screen. They are currently working with me on Wallace, and it's been a great experience working with them as well. So it's not just me. And on the right, you will see a nice chrono sequence of the Anderson Lab. And on the bottom, you'll see our sponsors, which are many, and affiliations. So thank you, and thus ends the presentation.
Thanks, Jamie. So thanks a lot, Jamie, for the presentation and for all the hard work. That's an exciting set of tools.
Thank you.
Everybody, please send your questions for Jamie so we can get his responses while we're all online and communicating. Jamie, I've got two questions from Luis Escobar, who's an assistant professor at Virginia Tech. First question, Why do you propose using those 6 combinations of maxent features, linear, quadratic, hinge, kind of why that choice of feature combinations?
We elected not to include threshold and product features. For threshold, Stephen Phillips, who's the developer of maxent himself, is advocating for people not to use that because he feels like it results in very abrupt responses that are not kind of ecologically relevant. And for product, it's hard to interpret what the results mean when there are lots of product features. Sometimes people find them useful, but we wanted to keep it relatively simple and also to have responses that look more similar to kind of like GLMs and GAMs than not. And so that's why we decided to go with those particular features. But we're very open to changing this at any point if people have arguments against it.
I think these are questions just to understand rather than to argue. But second question, Luis asks, what evaluation metric do you believe is the most biologically realistic from those available within Wallace?
That's a loaded question. So thank you, Luis. I am not qualified to answer what is the best anything. And I think that kind of also, that wouldn't be in line with what we're trying to do here. We're just trying to provide available options and have the user decide. So yeah, I really don't know how to answer which is the most ecologically relevant evaluation stat. Maybe none of them. Maybe all of them. I'd be curious to know what Luis thinks.
Well, Luis, I know you're listening. You're welcome to flip me a quick email. I'm a co-author with Luis on a recent paper on Darwin's foxes that kind of explored a bunch of evaluation metrics.
Yes, it's a very interesting paper, by the way. I read it.
Well, I'm sure that's what Luis is thinking about.
Yes, and so it would be great to include more evaluation metrics that people think are more closely related to the ecology.
I have a question here from the group here at KU, Marlon Cobos.
Have you ever had thinking about what's better to use to choose models, the delta AAC value or the omission rates? Have you ever had cases in which the delta AAC, the lowest delta AAC value had non-significant models or something like that?
Yeah, so E&M eval reports the AIC value, and it also reports emission rates in different iterations of AUC. The biggest difference, in my opinion, between AIC and the other ones is that AIC does not use cross-validation stats to calculate its value. So all the spatial cross-validation we went through that we thought was very useful for evaluating the model, AIC does not incorporate any of that. It's just the likelihood. And then it has some variable complexity penalization, right? So AUC and emission rate and the other stats that might be calculated via the partitions, I personally like better because I think that partitioning spatially is a better way to evaluate models than just looking at a single value, which would be AIC. But a lot of people might have different opinions, and that's why we offer you the entire table and allow you to pick. And some people might even combine the two and look at all the stats together to decide what's optimal. I don't know if I answered your question.
Yeah, it's okay. It's okay. I think the same way than you.
Yeah.
And by the way, I should be fair to Luis Escobar. His two questions were prefaced by action. The Wallace platform seems very complete and friendly. So
No, I know Luis, and he is one of the friendliest people I've ever met. So I had no impression that those questions were aggressive at all.
I guess I have a question for you, which is, kind of the dimensions and the parameters that you explore when you do these model selection exercises. We've kind of become impressed with another dimension, which is essentially the set of environmental variables to do the model fitting.
Mm-hmm.
And, frequently what do? We, reduce by redundancy, and then maybe use a jackknife or something like that to throw out a few more. Mm-hmm. And you always end up with this question of how many should I throw out?
Right.
But I'm just wondering, so you've got the response type and you've got the regularization parameter.
Right.
We could probably throw in environmental data sets, mm-hmm. but are there other parameters that would be interesting to explore as far as including them and excluding them or varying them in these exercises?
Right. Yeah. We talked about possibly including some step where you're able to remove environmental variables based on correlations and things. A lot of people have many different techniques in order to limit the number of variables that go into the model. But then again, there are two camps, one which wants to remove variables from the offset, and the other which kind of lets Maxent do the work of removing the variables. But yeah, I mean, we like to include all the options that make sense to the most people. It's just that our lab mostly is in the other camp, where we just kind of give Maxent, unless there's very strong evidence that we have that the species responds particularly to a couple of variables, we just give it all the information we think might be useful and let Maxent do the work. But yeah, there's a whole other camp that feels a different way, that wants to limit the variables that go in. And actually, our lab came out with a paper recently where we did that, where we had expert decisions used to limit the number of variables that went into the model. And for that particular study, we actually came out with very good models. So, yeah.
Well, that's interesting. I guess so. It's strictly speaking when the variable has zero contribution to the model.
Yeah, for Maxent at least. Right, and the higher you pump up the regularization multiplier, the more variables get tossed out, and so that's why we emphasize you should look at a number of different models over this range in order to see kind of which is the best model for you. And we also encourage you not to just look at numbers, but to look at the map, to look at the response curves. So look at a lot of different things in order to decide what's your optimal model, not just the numbers.
Question?
Yeah.
Here we have a question from Ali, a student of mine.
Hello.
Thank you very much for the presentation. I have a question. It's kind of unrelated to the technical part that people were asking, and also it was kind of 10 minutes late, so probably you explained that at the beginning. But my question is that I guess this platform kind of needs the highest speed internet. So for countries with, probably, I'm not sure. But do you have kind of offline version of this platform that for countries with lower speed of the internet, people can kind of download them and they use it, or it should be definitely online? And also for the problems, if they find some kind of problems in the modeling process, how they can edit the bots, or how they can send the feedback to you.
So for the first question, everything I did was offline. So
it was offline.
Right. Wallace is, I mentioned the words web app because Shiny is really meant for building web apps, but we're using it just to make the GUI. So all of this stuff is offline. It's on your local machine. In the future, we'd like to put it up on a website once we have a dedicated server, but we're not quite there yet. We're working on that. But yeah, this is all offline, so it just depends on the speed of your computer. However, if you want to download things like GBIF data, or you want to download the WorldCom data, you would need an internet connection. But we have options so that if you have your own data, you can input it and not have to use an internet connection at all. As for the, I'm sorry, the second question again was, oh, regarding the problems with the modeling?
Yeah, I guess it was, you explained through the GitHub, they can send the feedback, right?
Yes.
For the problems that related to the modeling process. I mean, for the zone that they choose, I mean, sometimes should be, I mean, it will be wider based on the parameters that you use for kind of optimizing the model. So
right. So there are two main kinds of questions I'd expect. One is bugs, which are software bugs. So the application crashes. Those are the kinds of things I think would be best to go to GitHub. And the other ones are methodological or philosophical issues where, my model looks funny. What does this mean? That kind of stuff would go maybe to the Wallace Google group, or even to there's a very well-attended Maxent Google group that addresses SDM issues, and those kinds of questions can go there too. But the other thing is Wallace made my model look funny. Why did it do that? That might be a bug too. And so yeah, there are things that might be in the middle. But yeah, there are multiple places to put your issue, and don't feel any pressure if you don't know where to put it. If I think it's the wrong place, I'll just put it the other place. But yeah.
That's awesome. Thank you.
Yeah.
Okay, well, Jamie, I think that's all the questions we have. So thank you a million times over, and thanks to you and your co-authors for developing a neat open access, open source platform that can be very fundamental for the whole community.
Yes, thank you so much for having me. This is a really good time. And I encourage anyone who has more feedback to email our group with questions.
Great. Thanks again, Jamie.